Louping ill and tick-borne encephalitis. So Flaviviridae has the flavivirus genus. A single-strand RNA, it's enveloped, which means it has weak resistance. It needs a tick or mosquito as a vector. This makes them arboviruses. They're zoonotic. They have a uroxin host range, and the geographical location is characteristic to each disease. Certain viruses live in certain areas. They're not worldwide. For louping ill, the occurrence is in the British Isles, Scandinavia, and the Mediterranean regions such as Turkey, Greece, and Spain. For the etiology, the louping ill virus is a part of the flavivirus genus. For the epidemiology, the vector is Ixodes ricinus, which is the castor bean tick. They are the biological vector and they allow transovarial transmission. There are urixin species, but it occurs mainly in sheep, but it also occurs in birds, cattle, goats, and foals. And humans as well. It's a zoonotic infection, but it's rare. The virus is shed in the milk, so it affects young animals. For the pathogenesis, a tick inoculates the virus into the blood, and a viremia occurs, and the primary replication happens in the lymph nodes, and then it spreads to the organs. A second viremia occurs, and it spreads to the central nervous system, and this causes meningoencephalitis. For the clinical signs, the incubation period is 1 to 3 weeks. In sheep, it causes biphasic fever, and the central nervous system signs are incoordination, ataxia, they're in the jump over fence stance, and depression and anorexia. In the pathology, we can see no macroscopic lesions, but in the histopathology, we can see lymphocytic infiltration, neuron necrosis and apoptosis, and glial cell proliferation. The differential diagnosis is rabies, listeriosis, and scabies. For the diagnosis, we look at the epidemiology, the clinical signs, and the post-mortem lesions. To detect the virus directly, we do real-time PCR, and we can do isolation with IF and immunohistochemistry. To detect the virus indirectly, we do ELISA, viral neutralization, and HIA is the main one. In terms of public health, human infection is rare, but it can cause fever and meningoencephalitis. For prevention and control, you need to control the number of ticks, and there's also an inactivated vaccine in endemic regions. Tick-borne encephalitis is a human disease. It causes an asymptomatic infection in animals, but the source of infection is milk or ticks. For the occurrence, it occurs in Central Europe. It's endemic. It causes natural foci infections. And there's also lineages in Asia and Far Eastern countries. For the etiology, the tick-borne encephalitis virus is part of the flavivirus genus. And for the epidemiology, the vector is Ixodes ricinus. It's seasonal. This is the castor bean tick. It's a biological vector and it causes transovarial transmission. The virus is urixin. It infects humans, but the virus is maintained in mammals and birds. The virus is shed in milk. Goat milk is the source, especially raw goat milk. The natural cycle is between rodents, small mammals, these are carriers, and the ticks. For the pathogenesis of the disease, so a tick bite or consuming raw milk causes local multiplication, then a viremia occurs, and then the secondary replication happens in the central nervous system. For the clinical signs, in animals it's subclinical, however young animals can get depression, ataxia and convulsions. In humans it can also be subclinical as well. Or they can get biphasic fever, so fever with flu-like symptoms and fever with central nervous system signs. There is usually a complete recovery, however permanent lesions can remain. For the diagnosis, we look at the epidemiology, clinical signs and post-mortem lesions. To detect the virus directly, we do real-time PCR and we do isolation, IF and immunohistochemistry. To detect the virus indirectly, we do ELISA, viral neutralization and HAI. For prevention and control, we do tick control and there's also an inactivated vaccine. There is a long-lasting immunity. It lasts 3 to 4 years.